Okay, welcome back for Intro to Algorithms Part 6. This is the last of our algorithms sections, and we will finally be getting to where we count the number of occurrences of patterns that don't have exact matches. So you'll want to go ahead and download the PowerPoint from Canvas, and I will go ahead and start sharing. So if you're following along in the textbooks, which you should be, if you really want to get the full benefit of the conceptual side of what's going on here, what we've done so far, right? We started out finding exact pattern matches and counting the frequency of those words to try to find hidden messages, right? And in principle, these are transcription factor binding sites, or in the specific case, they are DNA A boxes. These are the proteins that mediate bacterial replication. And then we moved to the situation where we had to not only look in a particular part of the genome, right? We were started out given a 500 base pair region and then define, what are the most frequent k-mers in that region. Well, then we moved to, okay, you have to scan across the whole genome and find frequent words that are clumped together. And the whole idea there is, how many times do you expect a k-mer of length 9, for example, to occur just by random chance? And then, so we were given sort of the, it was defined as T, where now we're no longer looking for the maximum count. We're looking at words that occur more than a certain number of times. And that's typically defined by the expectation for a random k-mer of that length. And so we want to find it more times than that. Then we discovered that if you do that on a whole genome, you actually find lots of clumped k-mers. And so we learned to look at or take into account more of the biology. So we did the minimum skew problem, and that's what I talked about last time, and we plotted the difference in number of G's minus the number of C's to look at where you get that inflection point, and that represents right, the difference in mutation rates for single-stranded DNA, which tends to deaminate the cytosines to become thymines, and so you get this inflection point where it stops decreasing in that G minus C number and then starts increasing, and that is our region where we think replication ought to start, and then we can look in that region for frequent words. So this example from Vibrio, right? That was it's a big help if you do that analysis on Vibrio, and it turns out that there are actually more DNA binding sites than you get with the exact match algorithm if you allow for mismatches. And so that's where we're headed today. How do we find, or how do we include the frequency of these patterns if we include or account for the fact that you don't have to have exact sequence matches, right? Because again, this is a protein DNA interaction, which doesn't rely on specific sequence complementarity. So the last four are what we're going to deal with today. And I guess I should also say, right, in the previous part, we did the GC counting part, but then there were two other problems, 18 and 19, that you will need to have done to complete the stuff for today. That was being able to compute the Hamming distance, so how many different two sequences, and then how to generate all of the permutations of a pattern that are some distance away. So, you know, a Hamming distance d equals 1 means you can have a substitution of any nucleotide at any position in your pattern. How do you generate that whole suite of k-mers? Let me expand that. Swap displays. Okay, so the first problem here, find an approximate occurrence of pattern in a string. Let's go ahead and look at Rosalind. So what are we going to be given? Rosalind is going to give us a pattern and then some text, right? The genome sequence, and then an integer d. So that is the Hamming distance that we're supposed to allow. So what we need to do is take this pattern and compare it to all the windows along text and count it if it is 3 or less mismatches away. And then the output that it's looking for is the start position for each one of those matches. So presumably if we were to slide along here at 0, 1, 2, 3, 4, 5, 6, so right here, there should be a match that is 3 or less. Okay. So how does that work? Right, I just have a cartoon example here. So this is really what we need to write some code to do. Whoops. The text here, I should have, so this top row is the text, and then AAC would be like the pattern, right? I've just simplified this example, so I'm saying the pattern that we're given is AAC, and in this case, instead of D being 3, say D is 1. And I've just done this to sort of simplify the example so that you can think about how does this algorithm need to work. So we've got our text, we've got our pattern, and we just say, okay, AAC matched that first window. D is 0, right? There are no mismatches. And how could we estimate that? We've written a function, I think it's number 18, that computes the distance for us. So we could just send, say we've got this as our pattern and this as our sequence window, send those two things into the Hamming distance function, and it should return a 0. Okay, then we need to slide over one so that we have ACA as our window in the text, and then we compare that again to AAC in the pattern, and now there are two mismatches, right? We mismatch at the C and we mismatch at the A there. So that's D equals 2. We wouldn't want to do anything with that, right? Because D is less than 1, that's going to be false. Okay, then we slide along and get the next one. Again, we have two mismatches, so D is 2, don't want to do anything. And then on the last one, AAC, actually this is only a distance of 1, right? We only have a mismatch at the last position, so that will evaluate to true, D less than or equal to 1, true. And so we have position 3, right? And so our answer for Rosalind in this example, right, it wants the start positions would be 0 and 3. All right. So what does that look like in terms of, you know, now let's move to conceptually, what is it, to something that looks a little bit like pseudocode, at least an outline of our algorithm. So we're going to, you know, you don't have to define these as functions. In this case, you're really going to want to, because you're going to want to modify this code so that it can actually count instead of return k-mers. And I'm saying that, you know, just to a little foreshadowing that you're going to need to be able to count the patterns with mismatches later for 21. So let's define a function that it takes pattern, text, and D, right? Pattern, text, D. Right, so that's your with open, and then you have data read lines for that one, and that one, and this one has to be an integer, right? So don't forget to make D an integer. Then we're going to initiate a list to hold the positions. You can name that whatever you want, but it's just an empty list so that we can return, right, this list. Okay, obviously it's not printed like a list, it's printed with the well, these are integers, so right, we're going to use the asterisk when we end up needing to print that, but that's our list of positions. Okay, then we need to do a sliding window, right? We're going to do a sliding window across the text to get each thing that we need to compare to our pattern. Then we send the pattern or the K-mer, right, that Rosalind gave us, and the current window, and have our Hamming distance function compute D. Right, so that's what I was saying. So in this first case, the K-mer or pattern, whatever you want to call it, is AAC. So we send that and call it sequence 1 if you want, and then AAC, the first window in our sliding window, call that sequence 2. And remember the Hamming distance function, if you go back to 18, it takes 2 sequences. So those are our 2 sequences that we're going to feed into the Hamming distance function. And that Hamming distance function returns a number that is D, right? So it tells us how many mismatches are there. So then we ask, is the Hamming distance less than or equal to D? If it is true, then we append that current start position to the list of the positions, right? Remember, the start position is taken from our sliding window. So we append the current window start position to our list of positions and then return the list of positions. So this is, again, this is exactly what I just walked through here. This is just something closer to pseudocode that does the same thing. So that should be enough to solve number 20. Then you will want to modify that code so that instead of returning the list of positions, right? So instead of appending the current window start position, you just want to add one to some counter, right? So instead of initiating a list where we hold positions, we want to just have a count. And then if the Hamming distance is less than or equal to D, we're going to add one to that current K-mer that we're counting matches on. And then you'll want to name that function something like count K-mers with mismatches or something like that. All right, so number 21. Let's go to Rosalind and look at 21. In this case, now we're to the point where we're going to count frequent words that have mismatches. So this is very much like number 10 or number 11. Well, at least in terms of what we're trying to do, number 11, find the most frequent words in a string. This problem, number 21, is find the most frequent words in a string with mismatches. And the tricky thing here that makes it unlike that initial solution, and not even just a straightforward extension of number 20, is that in number 20, Rosalind gave us the pattern that we have to look at. But what happens if you allow for mismatches in the counting is that the actual pattern might not occur in the text. So what do I mean by that? So here's our string, right? So I've just grabbed a pretend string here, or an example. It's not pretend. And then in this case, we're going to look at k equals 5. And just to go back, just so we're clear, right, the sample data, we're going to be given text, we're going to be given k, and we're going to be given the d, the distance that we want to allow. And then what do we want to output? We want the k-mers that fit that criteria. So we have 4 mers, 4 base pair patterns, and each of these must be happening in this text with 1 mismatch, okay, and frequently. So those are the 3 most frequent. Okay, so in my example, I have a text here and my k-mer is 5, and we're going to allow for d equals 1 mismatch. So I already know from this example, and I did this just to try to show what's happening, is that the most frequent k-mer in this string with d equal 1 mismatch is a string of 5 A's. The string of 5 A's doesn't occur exactly in the string anywhere, right? In fact, there is no 5 base pair pattern that occurs more than 1 time in this string. So if it doesn't occur in the string, how do we count it? Because in all of the examples that we've done up to this point, in order to count the pattern, we had to be given the pattern. And you know, if you go back to number 11, you had to slide along and get windows and say, okay, that's my first pattern, then slide along, okay, that's my second pattern. Well, if you did that and slid along and got all the 5 base pair patterns out of this string, you would not be able to find the k-mer that occurs the most frequently if you allow for mismatches, right? Because the most frequent one I've already shown is 5 A's. All right, so how can we find that? Well, we have to use our neighbors function. So now thinking about how we're going to do this, we are going to get that first window, AACAA. So that's the first k-mer we need to count. Okay, but we don't need to just count that one. We need to generate all the k-mers that are 1 mismatch away. So we can just send, right? This is exactly how it ought to look. If my function was called neighborhood, so from which number is it? Number 19. Yeah, so if we generate the d neighborhood of a string, so if we have the function from this problem called neighbors, it's going to take a pattern. It's going to take a pattern and a distance and generate all of the k-mers that are, in this case, 1 mismatch away. Then we'll be able to use all of these and count how frequent are these in the text. And, well, how many times are they in the text with a d of 1? Okay, right? Because if we just counted them exactly in the text, you would find some of these don't ever occur, but we want to count them if they're 1 base pair, 1 mismatch away. Then, okay, so we do that for the first window, then we slide down to the next window, okay? And we do the same thing, generate all the 1 mismatch away k-mers, count them, and keep on doing that. So we have to slide all the way down. So essentially it's just like number 11, except that we have way more k-mers that we need to look at because we're going to allow for the mismatches. And it introduces this intermediate step where we can't just check the current window. We have to first generate all the neighboring k-mers that are d distance away and then count. Okay, so that we include all the searches that might be d distance away. And then from an algorithmic standpoint, that's a lot of k-mers potentially that you're going to have to count, or that the computer's going to have to count. And so we want to try to avoid counting the same k-mer twice. So in the first window, AACAA, right, we get the neighborhood and it has this ATCAA k-mer. Well, when we get over to this pattern, I don't know which index value that is, but it also generates ATCAA. We don't need to count this one again because we already counted it from this neighborhood. Okay, so in our algorithm, we'll want to check to see, did we already count this thing? And if we did, then don't bother counting it again, right? So putting in a little more pseudocode terms, you say, right, if this thing is already in our dictionary, don't count it. Okay. All right, so what does this look like in terms of an outline at least? So we're going to define a function that takes text k and d. Got to go to 21. So it'll take text k and d, right? And you'll have to do the trick where you read this line in and then split it. And then we'll want to initialize a frequency dictionary to hold the k-mers. Okay, and if if you've already solved some of those earlier problems, we talked a lot in class last time about how to employ the dictionary method for counting. And that was problem number 16. So you've initialized a frequency dictionary, right? Because that is, where did Rosalind go? These are going to be the keys for the dictionary, right? So in our dictionary solution for counting k-mers, we have the key, which is the pattern, and then the value for that key is how many times did it occur when we counted it. Right, so we're going to end up returning the keys here. So we make a sliding window to get each k-mer. Those k-mers are the patterns, whatever you want to call it. We've been calling it patterns most of the time up to this point, but I've sort of slipped back into k-mer terminology, which is the more common way to think about it. It's just that Rosalind often calls them patterns. So these are synonymous. So we get that first, right? So this is what I'm describing. We get the first k-mer and then we send it to the neighborhood. So sliding window, we get the pattern, we generate a list of k-mers that are less than or equal to d distance from the current k-mer, right? We can generate that whole thing by a sliding window that sends each one to our neighborhood function. Okay, then for each k-mer in the neighborhood list, right? So we're going to slide along our neighborhood list that we just got. If the k-mer is not already in the frequency dictionary. So we saw something like this in problem 16. except in problem 16, we also had to test whether it occurred, right? We had something like if the k-mer is, or if the value is less than or equal to t and it's not in the dictionary already, then do something. Okay, in this case, if the k-mer is not already in the dictionary, then go ahead and count it. And this is where you need to have done the modification of number 20 that will count, because right, the simplest way to do this is take your k-mer, if it's not already in the dictionary, send it to the count with mismatches function from number 20, and then that becomes the value for the current k-mer key. Right, so this is how you specify, and right, if it's not already in the dictionary, this is going to put the current k-mer in the dictionary and assign it a value here that equals how many times that thing occurred with d less than or equal to d mismatches. Okay, so once this will generate the whole dictionary. Then we just need to get the maximum value from that dictionary. Again, showed how to do that in 16. And then whoops, I keep doing that. We return the list of keys that match the maximum value. So this part we've kind of already done. You should have some code that you can pull from 16 that will do something like this, except we're back to finding the maximum value and pulling out the ones that are the maximum value rather than comparing it against the value t. Remember that's the clumping problem, number 16. All right, so the last step is that we need to not only find the most frequent words with mismatches in the string, but right, if you look back at, let's jump to here, right? Some of these are reverse complements of the others. So it's not enough to just count one strand of DNA, because some of these binding sites are on the other strand. So in order to get to the last problem, which is to find the mismatches and reverse complements, we need to first get the reverse complement of a string. This one should be easy, right? If you go to your beginning bioinformatics tutorial, part 6, that's the one where we use BioPython. BioPython has a function for reverse complement. So you should have somewhere along the way already done number 22. It's just going to be a matter of solving it for the Rosalind thing. It's going to give you a string. You're going to use BioPython to get reverse complement of that string. And there should be, this one should be quick and easy. And number 23, also not a big stretch. The only difference here is that we now need to include reverse complements for all of those patterns that we checked. Okay. So if you think about what we did here, right, when we were checking all these patterns, we need to take the reverse, whoops. So for example, AATAA, we also need to check TTATT. And we're going to think, okay, that the reverse complement of this thing, we're going to add the count, how many times of TTATT occurs to this one, and then put that in the dictionary. So how are you solving this? This is, whoops, this is the very high level, is that we just need to incorporate number 22, that is the reverse complement, into our solution for 21. So what does that look like? Well, for each neighborhood, remember here we were generating the neighborhoods in this part of number 21. So for each neighborhood, if it's not already in the frequency dictionary, we go ahead and count the number of occurrences using our function, modified function from number 20. Okay, that is exactly what we did in 21, problem 21. But now we need to add the step to get the reverse complement of the k-mer that we just counted. Count the occurrences of that reverse complement k-mer, and then add those two counts together before we add it to the dictionary k-mer value. Okay. So hopefully that makes sense. All we're doing is adding a step to get the reverse complement, count it again, then add those two values together as the actual value for whatever our current k-mer that we're counting in the neighborhood list. Okay, that potentially that could be a huge number of k-mers, but because we're doing that check of if it's not already in the dictionary, we won't count any of them twice. So at least that should speed things up. Okay, and then the last two steps are the same. Just get the max value and return the list of keys that match that maximum value. And that's it. So that's pretty much it for the programming. If you look at E. coli, which was our sort of problematic one, the DNA boxes, there are actually 4 of them if you allow for a mismatch. And at least in principle, this would be the origin of replication in E. coli. You would want to do some wet lab experiences maybe to, make a deletion in here and see if you stop replication, something like that, just to confirm. But computationally, in principle, we've discovered where E. coli starts replicating. This is an active area of research, like using computational methods to find origins of replication. These slides are from the maybe 2nd edition of the textbook, and they had these. I assume people who had agreed to be contacted if you were interested in doing this kind of research, but you get cases where it's not so clear where the minimum skew is. And so some people think that means that those species of bacteria have multiple origins of replication. Okay. So the open problem is whether you can solve this computationally, or whether this requires, it may just require wet lab experiments, right? It does happen that there's just not enough information in the sequence itself to tell you, or at least not that we can recognize. In principle, we should be able to, right? If you think of the DNA as just the sort of software program for the cell, then we should be able to write some software that recognizes how the cell understands its own software. Okay. So in principle, you might be able to solve this, but it has not been solved, at least as far as I know, and as far as the last version of the textbook. It gets more complicated in Archaea. We know that Archaea have multiple origins of replication. Okay, and so far there's no algorithm, at least as of 2nd printing of the textbook, to confirm origins of replication in Archaea. Right, because you can find them, the more of these min skews, right? So how do you define min skew in this? Well, you could just do it at, whoops. scale the inflection points somehow. Okay, so your inflection point has to be over a certain range or with a certain minimum and maximum over a given range to be defined as an origin of replication, but then you've got to count frequent words in those. And what happens is sometimes they don't match, right? You get a different word for each origin of replication. So lots of issues, interesting issues, I would say, in defining origins of replication that way. And then you get into things that have multiple chromosomes. So yeast, for example, have lots and lots of origins of replication. I think there actually might be a software tool for this now. And there's also been technology like ChIP-seq that has become more and more common. So if you have an DNA-A antibody, for example, you might be able to pull down sequences. But this, you know, trying to define origins of replication is still active. This is a bioRxiv preprint from last year. And because this is sort of the end of our algorithm section, if you're interested in transcription finder binding sites more generally, and how do you define those, you can continue on in the textbook. Chapters 2 and 3 go into even more depth about finding motifs in DNA. So a lot of times if you're reading literature about transcription factor binding sites, you'll see these motif logos, and the height of the letter is how conserved is that position in the transcription factor binding site. So for example, in this position 2, 4, 6, 5, 6 through 9, it's always that base pair. Otherwise, the DNA-A doesn't bind. And this is for Deinococcus radiodurans. There's a little background pasted over on the left. This is a highly radioactive, radiation-resistant bacteria, and so it gets lots of attention for how does it do? How is it resistant to radiation? This green one is the sort of canonical DNA box, but they don't, all of the DNA boxes don't match in this bacteria, and so you end up with seeing that position 1 and 3 are both variable. Position 3 can be an A or a T. Position 1 can be a T, A, or C, or G. I guess the yellow one is too squished to even see, but it's a G. So active, interesting area of research, you know, thinking even more broadly. I think I mentioned this maybe in class before, one of the early videos on algorithms. This is extendable to, you know, if you have RNA-seq data and you have co-regulatory networks, you might then ask if there are any hidden messages in the upstream region of all the genes you know are co-regulated, right? There's an example here. I didn't stick it into the PowerPoint, but this is one that my lab has been interested in, and we may do this as one of the projects. What these guys found is that dosage compensation of the sex chromosome is mediated by a transposon that sort of lands all over the X chromosome and brings with it things that are close to the dosage compensation entry sites. I mean, you can think of those as transcription factor binding sites. And so they identified the mechanism of dosage compensation here, or at least the spread of dosage compensation, using some methods that are not far off from what we just did with problem 23. All right. So that's it for algorithms. Happy coding.